let's start. Otherwise, as Elad said, I forget to thank all the people involved in this work. So, from our everyday life experience, so all we know that our brains move between two global states. We move from a conscious to an unconscious condition, which are occurring when we are awake and then fall asleep, or the other way around. Okay. In these two global states, cortical networks display different and specific patterns of activity. And referring to a classic review on this topic, which is shown here in this slide, you can see that on the left, single neurons, when the brain is in this conscious state, so when it is awake, display this pattern of spikes, which are rather irregular. And if you move at a larger scale of the EEG, during awake, the EEG appears to be very noisy, so desynchronized. For this reason, this awake state, let's say, or conscious state is also often called desynchronized. But when the cat, in this case, moves towards the fall asleep, you start to see in the brain something which is a hallmark of this condition, where you have a transition at the single level neuron between a silent period and the active period, which are referred often as the down states and the up states. This alternation between a microscopic state observable at the level of single neuron is also visible at the level of the macro scale, at the level of EEG. And they are organized in not only at the local level, but they are organized as a chain of activation and inactivation that in the brain occur as a slow wave. For this reason, the desynchronized state is often named as slow wave activity, because you have slow activation wavefronts traveling across the cortex, as shown in this video. The different phases of the wave are shown with different color codes, and you can have a very complex spatial temporal pattern, moving from, for instance, the spiral waves observed on the top, and the planar waves you can see at the bottom. Okay, but this slow wave activity has been suggested by several of us as a kind of default activity pattern, because it seems to be occurring whenever you have a cortical network which is isolated. And you can, from a theoretical perspective, you can describe this condition simply relying to two main features of cortical networks, which is let's say the level of excitation these networks are receiving, and the level of fatigue, which can be both short-term depression or spike frequency adaptation. And so whenever you are sleeping, you are in the yellow region of this bifurcation diagram, and the fact that you are sleeping and you stay asleep actually means that this seems to be a rather attractive global state. Okay, this is the reason why we show this two valleys picture of this effective aniline engines. And whenever you change these parameters that are occurring thanks to changes in neuromodulation in the brain, then you move from a condition which you are very close to a pathological condition, like the one having a bar suppression with very long down states with sparse up states, and you move then to a quite regular alternation between up and down. And then when you cross the border, you move towards the desynchronized state, reminiscent of the awake life, you start to see this synchronized state, as I told you. But even if here you can imagine that these states are rather stereotyped, something is known from the literature, something more than something is known from the literature. And what is known that during the movement of this system from this desynchronized state towards the synchronized states, some features of the network, functional and structural, seems to change gradually, I would say. And for instance, at the macro scale, as also saw yesterday, there is at the level of in the interaction between different cortical areas. When you have a sedated brain, actually you see that the connectivity is sparse and not so strong as depicted by these matrices of correlation between cortical areas. While when you are awake on the left, you can see that this correlation matrix, the functional connectivity, starts to be stronger, much more shaped in some way. This is the same observed also in rats, as shown below, where you can see that these changes are so smooth because can be seen, for instance, under anesthesia, a different level of anesthesia. So there is a smoothing change of this correlation pattern. And if you move now at the micro scale on the right, you can see that these changes are also observable and appears in a rather peculiar way. When you look at what's happening in a cortical colony, in a specific cell assembly, homogeneous cell assembly around the layer 5. And in this case, when you, again, you have anesthetized rats, you can see that when you are under deep anesthesia, you have very long down states, in this case represented by this deflation of the local field potential, that when the anesthesia fades out, start to be more regular as in the middle, and then when the wakefulness is approached, the complexity arises in an alternation between patterns that we described as an alternation between microarousals and slow oscillation that are represented in what we described in this paper with Nuria Tortcolet. This alternation is among two different attractor states. So even if there are some smooth changes, the global behavior of this rhythm seems to change smoothly and at the same time very rapidly. So one question arises whether this slow wave activity can be considered as a monolithic state or rather is a spectrum of patterns, and how this spectrum of patterns are organized in order to prepare the cortical network to be awake. And this is what Antonio is going to show you.
Yes, hello. Can you hear me?
Yes, perfectly.
Okay, so I first have to shortly thank the organizers too, and I'm really happy to be able to present here, and I'm really enjoying the talks and the discussions. We here started from macro electrocorticographical recordings of a grid of electrodes spanning a large portion of the mouse brain, so several regions, and at different levels of isoflurane anesthesia. We are restricting basically to the region where slow oscillations are present, and as you see, these are clearly visible in the recordings, and they show with different levels of isoflurane different features. First of all, of course, the frequency. The frequency increases with lighter anesthesia. What we have done is that we have developed an entropy-based measure of this level of anesthesia and for the different levels of isoflurane. So the way we did this is to use the relative transition times channel-wise of the transition from the down to the up state, and through thanks to the computing the entropy, we basically measure the effective dimension of the space embedding these spatial temporal patterns traveling through the brain. This entropy index correlates well with the frequency of the waves, of the slow waves, and not only, but also with the CV of the interwave intervals and, for example, the down and up durations, and of course with the consultation of isoflurane, which is alone showed some variability depending on the subject variability, and also of course on other experimental conditions. So we had this measure with allowed us to analyze the different kind of complexities in this propagating spatial temporal patterns in the cortex. First of all, we analyzed the spatial complexity, so the basically the area occupied by the different modes of propagation. At a deeper level of anesthesia, we see that basically this space is occupied in two small regions, and going for to lighter and lighter levels of anesthesia, these regions increases and then shrinks again, apparently. This corresponds to for deeper level of anesthesia to seen in the grid of the electrode, so spatially, to basically two modes of propagation, as anterior to posterior and posterior to anterior. Whereas when the anesthesia gets lighter, then many more modes and more complex modes appear. This was quantified by computing, measuring both the kind of modes, number of modes of propagation, and their velocity. Here shown also on the so in a sketch of the mouse brain, we see that indeed at deep levels of anesthesia, we have basically two modes, anterior, posterior, and back. Whereas for lighter levels of anesthesia, we see the emergence of several modes, which are mainly going from medial to lateral and posterior. These are the length of these arrows also quantifies the velocity, which indeed increase for lighter anesthesia. In order to gain insights into the network mechanisms playing a role here, Andrea Galluzzi performed spiking neural network simulations of modules of recurrent networks, and using the external drive and a level of adaptation. Basically, using these two parameters, we set two working points corresponding to basically to different levels of excitability in the network, and retrieve basically the deep-like activity where the frequency of the waveforms are smaller, and a light-like situation with more with higher frequency and different modes. Indeed, in the complexity, in the entropy of these modes shows again for the deep-like, as in the experimental data, as two small region of occupation in the deep-like situation, and a much bigger and actually continuously changing region for the light-like. And this corresponds indeed to the two main modes in the deep-like, and a number of spontaneously emerging modes in the like situations, more complex. Now we thought of trying to quantify also the temporal complexity of the spatio-temporal patterns, and we did also because we observed that some of the recordings had, as in this example, non-stationarities. So apparently this was the case for some of them, and the way we also studied this emerging complexity or variability, we was by analyzing the entropy index in a time resolved manner. We observed that depending on the average value of this complexity, of this wave entropy, some of the data sets basically showed a change at the beginning, between the beginning and the end of experiments. And starting with a high level of entropy, this tended to increase even more, whereas for data sets that have a lower level of entropy, these levels tended to stay or even to lower even more. So we wanted to quantify these changes along the time, and by so the beginning of these data sets from the end, and we did this by fitting the distribution of these values from the beginning and for the end. And we saw indeed that this distribution tends to be different, and at the end of experiments, for the high levels of entropy, basically the peaks, the number of points tend to separate. So apparently there seems to be a kind of divergence and in the modes depending on the kind of state the network is at. So another feature was that when the entropy level is high, there were also more fluctuations. So this was, by the way, also the case for the simulations where we fit the two dynamical states. So our suggestion, and here I come to the conclusion, is that basically at the transition between the slow oscillation state and the asynchronous state, we are at a unstable point where the system basically has to decide whether to stay in the slow oscillations or to leave and go to the and awake. So this seems to be the attractive states, and our results are an indication that indeed the system, when it happens to reach this point, basically make a decision and go towards one of the of these more attractive, more stable dynamical states. So with this, I'm done, actually.